Imagine holding the power to rewrite life itself, to erase disease, to enhance strength, to design the future. What was once the realm of myth and science fiction is now real, coded within a microscopic pair of molecular scissors. It's called CRISPR, a tool so precise it can cut and paste DNA as easily as text on a screen. With it, scientists can edit the blueprint of life, changing the code that defines every living thing. But how does this technology work? And what happens when humanity begins editing its own story?
CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A mouthful, yes, but at its heart, it's nature's own editing system. It was discovered not in a lab, but in bacteria, where it evolved as a defense mechanism against invading viruses. When a virus attacked, the bacteria stored fragments of its enemy's DNA like a genetic memory. Next time that same virus invaded, the bacteria used these stored sequences to recognize and destroy it, a biological archive of past battles. Scientists realized they could harness this system to target and cut specific pieces of DNA in any organism, plants, animals, even humans. That discovery changed biology forever.
To understand CRISPR, imagine a high-tech word processor for genes. Every living cell carries its own version of a text document, DNA, made up of billions of letters, A, T, C, and G. CRISPR acts like a find and replace tool for that text. At its core, CRISPR relies on two key components: Cas9 enzyme, the molecular scissors that cut DNA; guide RNA, the GPS system that tells Cas9 exactly where to cut. When scientists design a guide RNA that matches a specific DNA sequence, Cas9 homes in and makes a clean cut. Once the DNA is cut, the cell's natural repair mechanisms spring into action, either patching it back together or inserting new genetic code. This gives scientists the ability to turn off genes, correct mutations, or even add entirely new traits. It's as if evolution itself has given us the edit button.
Today, CRISPR is transforming nearly every corner of biology. In medicine, it's being tested to treat genetic diseases once thought incurable, like sickle cell anemia, muscular dystrophy, and cystic fibrosis. Some patients have already seen life-changing results, their faulty genes corrected inside their own bodies. In agriculture, scientists are creating crops that resist drought, pests, and disease, with fewer chemicals and more nutrition. In conservation, CRISPR could help revive endangered species or control invasive ones that threaten ecosystems. But perhaps the most controversial frontier lies in human enhancement, editing embryos to remove disease, or even enhance intelligence, strength, or appearance. This is where science touches philosophy, and ethics become as important as biology.
CRISPR opens doors that are as terrifying as they are inspiring. If we can edit out disease, should we also edit out traits we don't like? Who decides what counts as an improvement? In 2018, the world was shaken when a Chinese scientist claimed to have created the first genetically edited babies. The backlash was immediate, not because the science failed, but because humanity wasn't ready. The line between healing and designing life suddenly blurred. Ethicists now warn that gene editing could deepen inequality, creating a future where the wealthy can afford genetic upgrades, while others are left behind. Yet others argue it's unethical not to use CRISPR. If it can save lives, why hold back? The truth is, CRISPR forces us to face an ancient question in a modern form. Just because we can, does it mean we should?
The next decade will define how humanity uses CRISPR. Already, scientists are developing new variations like CRISPR-Cas12, Cas13, and prime editing that are more accurate, safer, and capable of rewriting longer sequences of DNA. Imagine curing diseases before birth, growing organs for transplants, or erasing viruses from the human genome entirely. But the greatest potential may not be in changing life, it's in understanding it. CRISPR reveals that life's code is editable, but also precious, a reminder that we are both authors and characters in nature's story.
CRISPR is more than a tool. It's a mirror reflecting our deepest desires and fears. It shows us how far we've come from the first spark of life and how much responsibility comes with knowledge. In the end, the power to edit genes isn't just about science, it's about wisdom. Because the greatest experiment isn't in a lab, it's how we choose to shape the future of our own species. What do you think? Should humanity rewrite its genetic code or leave nature's script untouched? Share your thoughts in the comments and join us again as we continue decoding the science that shapes who we are. Subscribe to Gene Decode, where science meets simplicity and the mysteries of life become clear.